Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to talk about how sodium chloride is reabsorbed in the collecting ducts. So with that, let's give it a go. So before we talk about how sodium chloride is reabsorbed in the collecting ducts, let's talk about the main diagram that we're going to be using today. So on this diagram, we first have our epithelial cells that make up the wall of the nephron. These epithelial cells basically divide two compartments. The first compartment is the tubular lumen, which is present inside the nephron, and then we have the interstitial space, which is outside the nephron. Now we also have two sides of the epithelial cell. The first side of the membrane, right here, the membrane that faces the tubular lumen, is referred to as the apical membrane, whereas the membrane that faces the interstitial space is called the basolateral membrane. So now that we know what this diagram looks like, let's now talk about how sodium chloride is reabsorbed in our collecting ducts. So the first region that we're going to talk about is the cortical collecting duct. And we're going to start off with our principal cells, which are in the cortical collecting duct. So how do the principal cells deal with sodium chloride? Well, on the basolateral membrane, the principal cells are going to have the sodium-potassium pump. The sodium-potassium pump 3 sodium out of the cell into the interstitial space, and then 2 potassiums into the cell. And it accomplishes this by taking the energy from the hydrolysis of ATP and using that to pump both of these ions against their electrochemical gradients. This then brings us to the apical membrane. So on the apical membrane, we have epithelial sodium channels, or ENaC channels. So epithelial sodium channels are basically these channels on the apical membrane, and what they do is they allow sodium to move from the tubular lumen into the cell. Sodium moves down its electrochemical gradient, which was established by the sodium-potassium pump, into the cell. Now on the basolateral membrane, we have potassium channels. These potassium channels allow potassium to move out of the cell into the interstitial space, which helps to allow potassium to keep going through the sodium-potassium pump, allowing this pump to continue working. Now, in addition to these transcellular mechanisms, we also have a strong negative voltage inside the tubular lumen. This will basically repel chloride ions, which will allow chloride to move through paracellular routes into the interstitial space. So as a review, sodium comes into the cell through ENaC channels, and then once sodium is inside the cells, the sodium-potassium pump removes the sodium from the cell and brings it into the interstitial space. Chloride will move into the interstitial space through paracellular pathways, which is driven by the negative voltage in the tubular lumen. So that is basically how the principal cells work in the cortical collecting duct. So the next cell that we're going to look at in the cortical collecting duct is going to be the beta intercalated cells. So how do these cells deal with sodium chloride? Well, what these cells are going to do is, first of all, on the basolateral membrane, they are going to have a specific pump called VATPase. So what this pump does is it basically pumps hydronium ions into the interstitial space, and it does this by using the energy released from ATP hydrolysis. So this pump is going to be integral into how we reabsorb chloride through transcellular mechanisms, and we'll see how that works now. So inside the cell we have water, and water can dissociate into 2 ions. The first is going to be the hydronium ion, and the second is going to be the hydroxide ion. Now the hydronium ion is going to be pumped out of the cell through the VATPase. This will cause the reaction to move in the right direction, or in the forward direction, which will help to produce more hydronium and hydroxide ions, and this is because of Le Chatelier's principle. However, this hydroxide is then going to be used in further reactions. So inside the cell we have carbon dioxide, and the carbon dioxide will combine with hydroxide to form bicarbonate. And this reaction will be catalyzed by carbonic anhydrase. So once the bicarbonate is formed, it will basically be exchanged for chloride via the action of pendrin. So pendrin exchanges bicarbonate for chloride. Now, in addition to pendrin, we also have another transporter on the apical membrane. This transporter is NDCBE. So NDCBE basically allows 2 bicarbonates to come into the cell, 1 sodium to come into the cell, and 1 chloride to move out of the cell. So now that Pendrin brought in chloride into the cell, how does chloride move from the cell into the interstitial space? Well, one way it's going to be through the chloride channels. So the chloride channels allow chloride to move into the interstitial space, therefore allowing chloride reabsorption. So now that we talked about the cortical collecting duct, let's move to the intermedullary collecting duct and specifically look at the principal cells there. So how do the principal cells deal with sodium chloride? Well, on the basolateral membrane, we have the sodium-potassium pump. And as you know, it pumps 3 sodiums out, 2 potassiums in by using the energy released from ATP hydrolysis. So on the apical membrane, we basically have NaCl channels. And these NaCl channels allow sodium to flow down their electrochemical gradient from the tubule lumen into the cell. Now, in addition to NaCl channels, we also have CGGC channels. These are cyclic gated nucleotide channels, which basically allow sodium to flow down its electrochemical gradient into the cell. In addition, we also have sodium bicarbonate cotransporter. So what this does is it basically takes 1 sodium and 1 bicarbonate and moves it into the cell. So this is another way of sodium reabsorption in these principal cells in the intermedullary collecting duct. Now, on the basolateral membrane, we have potassium channels that allow potassium to move out of the cell and therefore helps to keep the sodium-potassium pump running. Now note that when the rate of sodium reabsorption is very high, the majority of chloride reabsorption in the intermedullary collecting duct will occur through the paracellular pathway. And the reason why is because when you're moving sodiums from the tubule lumen into the cell, negative charge starts to build up in the tubule lumen. As a result, this causes a negative voltage, which therefore allows chloride to move paracellularly into the interstitial space. Now, there is another mechanism by which how chloride will move into the cell, and this is going to be through an anion exchanger. So what this does is it basically takes an anion and exchanges it for chloride. Now, it's important to realize that the chloride that comes into the cell will move out of the cell through chloride channels into the interstitial space. Now, only 0 to 5% of chloride reabsorption occurs through this transcellular pathway. The majority of chloride reabsorption is going to take place through the paracellular pathway that we talked about already. So that is how sodium chloride is handled in the intermedullary collecting duct. I hope this video helped you understand how the collecting ducts basically handle sodium chloride, and I hope to see you in the next video. Thank you for watching, and I hope to see you next time.